Welcome to part 2. Let's keep on trucking. All right. Because diffusion depends on random particle motion. We're starting right here. Even when equilibrium is reached, particles of a solution will continue to move across the membrane in both directions. However, because almost equal numbers of particles move in each direction, there is no further change in concentration. Key checkpoint. What conditions are present when equilibrium is reached in the solution? Conditions that are present? Well, that means that the same amount of particles going one way are going the other way. And finally, on both sides of the membrane, the concentration is equal. So, diffusion. Please move with me to page 185. Now we're going to learn about a different kind of diffusion, one that is special, and one that is very important to biology. It's called osmosis. Although many substances can diffuse across biological membranes, some are too large or too strongly charged across the lipid bilayer. If a substance is able to diffuse across a membrane, the membrane is said to be permeable to it. Permeable means to allow access, to let something go through. A membrane is impermeable to substances that cannot pass across it. Most biological membranes are selectively permeable, meaning that some substances can pass across them and others cannot. Water passes quite easily across most membranes, even though many solute particles cannot. An important process known as osmosis is the result. Osmosis, and this is really important, osmosis is the diffusion of water through a selectively permeable membrane. Alright students, don't let osmosis fool you. It's not hard. It's just, you got to wrap your mind around the concept. So let's read about how osmosis works. Look at the beaker on the left in figure 715. There are more sugar molecules on the left side of the selectively permeable membrane than on the right side. That means that the concentration of water, and this is the trick, concentration of water. We sometimes don't think about the concentration of water because it's usually the solvent. But in this case, we need to think of water as in concentration of water. Is lower on the left side than it is on the right. The membrane is permeable to the water, but not the sugar. This means that the water can cross the membrane in both directions, but sugar cannot. As a result, there is a net movement of water, net means total, of water from the area of high concentration to the area of low concentration. Water will tend to move across the membrane until equilibrium is reached. At that point, the concentrations of water and sugar will be the same on both sides of the membrane. When this happens, the two solutions will be isotonic, which means same strength. When the experiment began, the more concentrated sugar solution was called hypertonic, which means above strength, as compared to the diluted sugar solution. The dilute sugar solution is hypotonic, means below strength. Alright, so here we've got a couple words. First, iso means the same, hyper means above strength. Other words like hyperactive, hyper, you know all kinds of, he's hyper, he's above strength, he's got too much energy. Hypo is under or below. For example, you know the word hypodermic needle, when the needle goes under your skin, into your vein, hypodermic needle. So let's check out this pretty cool little figure here. So osmosis. Okay, so we've got a selectively permeable membrane. Selectively means only a few things. Permeable means can cross. Membrane means barrier. So a selectively permeable membrane, and on one side we've got more sugar molecules than we do on the other side. And we have the same amount of water. But now we have to think of water as in concentration. This side has less water than this side, which has more water. As far as concentration goes, a higher concentration of water, a lower concentration of water. Why? Because there's more sugar on this side than there is on this side. Well, water will move across the membrane because water goes from water, like anything else, will diffuse from a higher concentration to a lower concentration. So water moves this way until the concentration of water is the same on both sides. This is wild. This is wicked cool. Look, it will move in and push the water up against gravity. Totally cool. Okay, please move with me to page 186. Okay, 186. I'm going to zoom out just a little. 186. All right, here's a little graph that'll help you. Great quiz question material. Isotonic. The concentration of the solutes is the same inside and outside the cell. So what that means is the same amount of water goes in as the same amount of water goes out. In our hypertonic, it means the solution has a higher solute concentration, more solute than the cell. So in this case, there is more solute outside. And what happens? Water goes out of the cell to balance out. And hypotonic means there is less solute outside the cell. What happens is water is going to rush in from outside the cell and cause the cell to expand. Now, let's keep reading. Osmotic pressure. For organisms to survive, they must have a way to balance the intake and loss of water. Osmosis exerts a pressure known as the osmotic pressure, on the hypertonic side, that means the side with less solute, of a selectively permeable membrane. Wait, let's go back. Remember our picture right here? In this case, which side here is the hypotonic side? Well, hopefully you will say that this side, hypotonic means below strength. Yeah, this side is the hypotonic side. So the osmotic pressure pushes this way. It is pushing that way. Okay, hypertonic side. So it pushes towards the hypertonic side of a selectively permeable membrane. Osmotic pressure can cause serious problems for a cell. Because the cell is filled with salts, sugars, proteins, and other molecules, it will almost always be hypertonic into fresh water. This means that osmotic pressure should produce a net movement of water into a typical cell that is surrounded by fresh water. If that happens, the volume of a cell will increase until the cell becomes swollen. Eventually, the cell may burst like an overinflated balloon. Unfortunately, the cells in large organisms are not in danger, are not in danger of bursting. Most cells in such organisms do not come in contact with fresh water. Instead, the cells are bathed in fluids, such as blood, that are isotonic, means they're the same concentration of water on both sides. Same concentration of solutes on both sides and water. These isotonic fluids have concentrations of dissolved materials roughly equal to those of the cells themselves. Other cells, such as plant cells and bacteria, which do come into contact with fresh water, are surrounded by a tough cell wall. Wow! Oh yeah! Function of the cell wall, to keep the cell from exploding when it comes into contact with fresh water. The cell wall prevents the cell from expanding, even under tremendous osmotic pressure. However, the increased osmotic pressure makes the cell extremely vulnerable to injuries to their cell walls. Checkpoint. What structures, checkpoint. What structures protect plant and bacterial cells from potential damage resulting from osmotic pressure? If you said the cell wall, you are a winner. All right. Let's continue to move on to the next page, 187. Stop.